Hi everyone, I'm Tyler Banks, a PhD student in Dr. Nayar's lab here at the university. In this presentation, I'll be introducing some of our studies related to computational neuroscience and discussing a recent research topic our lab has been pursuing. There's going to be a lot of content here that may get glossed over, so please feel free to ask questions at the end. I'll provide my contact info for anyone who'd like this video link. And again, this is a pre-recorded video, but I will be available for questions at the end. What is computational neuroscience? Computational neuroscience is a highly interdisciplinary field. It's technically a branch of neuroscience that uses mathematical models to abstract the functioning of brains, to integrate known information about the brain, and to understand basic principles of the nervous system like development or cognitive abilities. Ultimately, this research is used to design interventions to help alleviate neurological and neuropsychiatric illnesses like Parkinson's or depression, PTSD. So it goes without saying that to understand computational neuroscience, you'll need a bit of background in neuroscience itself. As you may know, the human brain is comprised of roughly 86 billion neurons or so, and over 100 trillion synapses, the connections between those neurons. Neurons like these shown here are comprised of semi-permeable membranes capable of allowing different types of charged ions in and out of the cell depending on a number of factors. However, they can be abstracted to that of just a simple integrator. They take inputs in the form of electrical stimulation on the dendritic end here at the top, and if enough stimulation is provided, they produce an all or nothing output on the axon end down at the bottom. These neurons are then coupled together, typically axon to dendrite, to form complex networks across your brain and even your entire body. Output from one cell to another can either excite the next cell, increasing its firing probability, or inhibit it, which reduces the probability. A network like this also sends electrical signals to peripheral components of the body, like your hand, to produce movement. And it's really mesmerizingly complex at times. So, as mentioned previously, we can abstract the neuron and primarily focus on the all or nothing output, also known as the action potential, shown top left right there. The cell only fires this action potential when enough current enters the cell body, causing a sudden burst of activity there at the base of the axon. The activity then travels to the end of the axon, signaling the next cell. And again, we can think of that cell body as a capacitor collecting charge from the input source, from, in this case, those dendrites collecting current from adjacent synapses, and action potentials as an on or off event. If we were to model a basic passive membrane that doesn't have a spiking mechanism, it's only accepting the current input, it looks something like the diagram here in the top left. In the top right, we have the biological inspiration, and again, at the top left is our modeled circuit, complete with a capacitor, leak from the channels from the cell membrane, because cells really aren't perfect maintainers of charge, and then our current source there on the left, which would be the other cells. This is then used to create differential equations based on things like Ohm's law and Kirchhoff's current law. So we can then extend our model by adding additional cellular dynamics like sodium and potassium pumps, those ions, on the semi-permeable membrane to model the action potential. And this is achieved mathematically with several additional differential equations, which are kind of beyond the scope of this. So the result of solving these equations is shown there at the bottom right, and then a real neuron is shown at the top. And even though these images have no biological data in common, you can see how remarkably similar they appear. Additional equations can be added for the synapse between cells, which are shown here, we can then simulate multiple cell types based on biological parameters collected in the lab, and then create networks to model a particular brain region. This is like the hippocampus or the amygdala. Factors like neurotransmitters, which alter the behaviors of the cells, or synapses, can also be introduced to increase complexity and realism. So my background is in computer science, and I didn't come from the neuroscience or even electrical engineering, so a lot of these concepts were new to me. Some of the things that weren't new were the concept of operating systems, software language, and solving difficult problems like differential equations using computers. And although we don't quite understand the operating system that our brain uses, our goal is to make progress towards understanding how billions of these individual neurons operate collectively, how they talk to each other, and implement functions, as well as thought. You may know of the National Academy of Engineering, and they have this Reverse Engineer the Brain Challenge as one of its 14 grand challenges. This will require engineers to collaborate closely with neuroscientists who solve a lot of these fascinating problems that we're seeing today. And really, this is a new field of neural engineering, so to say. Coming to the specifics, we make models of these neurons and then networks using the differential equations, and then solve them iteratively using techniques like the Euler approximation or the Runge-Kutta method. I'm probably not pronouncing that right. Luckily, by using existing software packages like Neuron, we can create neurons in a declarative manner and specify those attributes easily, as you can see in the middle there. These models can be run solving the equations on supercomputers or even your laptop. While the area has matured recently in years, many of the software solutions haven't. So part of my research includes simplifying that process for developing these models, and then putting the technology in the hands of the biologists in the lab who are actually seeing these cells live. And all of this is really cool, but how is any of it useful or practical in the real world? And the answer is computational neuroscience solves several different problems with performing experiments in a live biology lab. For one, neurons are incredibly small. They're from 4 to 100 microns in most cases. Performing experiments on individual cells is incredibly difficult, and perhaps even more importantly, it allows us to perform experiments on cells as if they were running in a living being, which you couldn't do without killing the thing. Gaining access to some of the cells deep in the brain can be difficult or impossible. We can then use computational neuroscience to simulate inputs to single cell types and a heterogeneous population, and then observe the effects in a controlled environment, which again is something that can't be done in the lab, or at least without some advanced optogenetics. All right, so here we are at some of the research we do at our lab. A lot of that stuff was a very brief introduction to computational neuroscience, and if you're interested in learning more about the basics of it, I definitely recommend Dr. Nair's 4590 Computational Neuroscience or corresponding graduate level course. Things are going to get a little bit more difficult from here. We're going to move right into some of the cutting edge research taking place again at our lab. In specific, I'm going to discuss our efforts to parse resonant and synchronizing mechanisms as they relate to intrinsic theta rhythm in the hippocampus. In previously published papers, our lab developed biophysical computational models of the hippocampus, which is the center of the brain thought to be responsible for the encoding and retrieval of memories. And these models were used to demonstrate several findings about the intrinsic mechanisms capable of generating a theta rhythm. In short, the neurotransmitter acetylcholine regulates memory encoding and retrieval by switching between a pattern separation and completion state. Excitatory recurrent connections inherently create seizure-like activity. Inhibitory basket cells of the CA3 alone fail to control the network stability. Inhibitory OLM cells and CA3, along with short-term depression in CA3 to CA3 synapses, play an important role in controlling stability, and high and low cholinergic states, which is the level of acetylcholine, require different stabilizing mechanisms to control the runaway excitation. In 2017, this model was extended to study theta oscillations in the 4 to 12 hertz range. 2017 paper addressed the question of why several cell structures were found to be critical for generating that theta rhythm in vitro, but then were not found to be critical in vivo. The main reason was the presence of multiple theta generator mechanisms, which sometimes substituted for one another. It was also found that low cholinergic states engaged the recurrent connections in generating theta, while high cholinergic states utilized the OLM pyramidal subnetwork, and that theta may be generated differently across states, and demonstrated a direct link between specific theta generators and neuromodulatory states. So the goal of this part of the presentation and upcoming paper is to categorize those intrinsic theta generating mechanisms found in the 2017 paper into resonant and synchronizing mechanisms. Resonant meaning the theta rhythm is generated as a product of the cellular and network dynamics, and synchronizing meaning that those elements that strengthen the mechanism. The most robust theta rhythms require at least one resonant and one synchronizing mechanism, and inactivation of these mechanisms is dependent on the generators and neuromodulatory states. So backing up a bit again, what is the meaning of a brain oscillation? So brain oscillations occur when groups of neurons fired in a synchronized manner and then it's recorded. It's known that the synchrony is important for cross-region communication between different areas of the brain. Again, this activity can be recorded by embedding electrodes into the brain or measuring at the scalp and then analyzing it as an EEG signal. If we look at this raster plot here at the top left, where the x-axis represents time and the y-axis represents the cell ID, and a dot represents a cell spike, you can see bands representing synchronized spiking. This information is then captured in the local field potential plot, or LFP, as an oscillation that can then be analyzed using digital signal processing tools. For example, a power spectral density that peaks at the oscillatory frequencies. The PSD is shown on the y-axis here in the plot. Theta oscillations, which emerge when groups of cells fire up at the same time, occur at about the 4 to 12 hertz range. An example of this where theta oscillations are known to occur is in the hippocampal place cells. In the experiment shown here, a rat moves around its environment, and certain cells have a favorite or preferred location that they're excited by. Plot B shows that the set of cells that were recorded here had a higher likelihood of firing when the rat moved towards the middle of the plane, shown at the top. This is how space is encoded in our brain as well. Sniffing patterns in rodents and the saccades in monkeys and humans also seem to have a prominent theta as recorded in those LFPs or EEG signals. So returning to our model network, here we have a schematic of our model brain region on the left. The entorhinal cortex provides input to the hippocampal pyramidal cells and basket cells in the CA3, as well as similar cells in the dendate gyrus. artificial input is introduced to the entorhinal cortex, causing the spike at roughly 15 Hertz Poisson without any kind of rhythmicity. We can focus on the intrinsic theta and then CA3. As you can see, the theta band oscillations emerge from the network as a result. And the big question is, of those cellular mechanisms that were found to be responsible for the emergence of theta in that 2017 paper mentioned previously, which ones are resonant and which ones are synchronizing? So a lot of the details, the technical implementation is provided in those previous papers, but in brief, the model utilized the Zizekevich formulation for its cells, which provides kind of a reduced order model that preserves a lot of the neurocomputational properties of the more biologically detailed models. The networks were scaled down from about 1.6 million neurons in the rat hippocampus to 557. We preserved the cell type ratios when doing so. Model cells were then distributed uniformly in 3D space, or simulated, separated into 3 regions: the CA3, the EC, and the DG regions, with the dimensions that approximate the respective dimensions of the hippocampus. The cells were then connected with respect to actual biological data. Synaptic currents modeled AMPA, NMDA, GABA A, GABA B, which are the excitatory and inhibitory synapses, as well as some of the short-term plasticity between those cells. To model the effects of the neurotransmitter acetylcholine on those synapses, AMPA synapses, which is the excitatory, the currents for those were scaled by an artificial value between 0 and 2 to correspond with high and low states of that neurotransmitter. To generate the histograms displaying the relative changes in theta, we took the relative power in the theta band and then divided by the total power, so the 4 to 12 Hertz range divided by the 0 to 50 Hertz, and then normalized the value of the full model run, and you'll see those later. Alright, so there's a lot going on this slide. By wielding that power of computational modeling, we can artificially isolate those regions of cells and then study their intrinsic dynamics. While doing this, it's important to maintain the basal firing rates of the CA3 pyramidal cells, and in some cases we did so by providing additional current injections as we would have done in the previous papers. So getting to the plots, here in experiment A, those pyramidal cells were isolated, and then each received a distinct train of Poisson input drawn from a log normal distribution. The power spectrum was then averaged over 100 simulations with different seeds. The oscillations recorded peaked in the theta range, and the reason for this is cells had the highest probability of spiking again only after the decay of the adaptation current in the synapse. In experiment B, pyramidal cells were disconnected from one another once again, only receiving input from the entorhinal cortex. Spike in the theta range also appeared organized despite the local connectivity. In experiment C, recurrent connections and spike frequency adaptation interacted to generate theta oscillations as well. Input was supplied directly to pyramidal cells once again because that EC pathway, which is the true input pathway, may have a separate role in generating theta. And again, theta was generated quite robustly here. There in experiment D, the pyramidal OLM interneuron subnetworks were found to generate theta through 2 mechanisms, which were discovered by varying the weight between those synapses. With weak synapses, OLM cells fired sparsely and sent inhibitory currents to the pyramidal cells at lower rates than theta frequency, and theta only emerged when those pyramidal cell spiking oscillations and sporadic OLM inhibition acted as a common input. With the strong synapses, OLM cells fired near theta frequency and then they generated theta locking inhibitory currents in that pyramidal cell network. And then finally, experiment C or E, the pyramidal to basket cells were isolated, and here it was found that the network didn't generate any rhythmic activity, initially due to short-term depression and the lower connection probability compared to the OLM subnetwork. By removing the short-term depression and then doubling the connectivity, oscillations were restored. Okay, so the last slide was kind of a mouthful. Here's kind of a brief overview for anybody watching the video later. So by running these experiments, we found several things that lent to the theta generation. The spike frequency adaptation, the slow inhibition, rhythmic external input, slow neuronal currents, as well as inhibitory feedback, non-rhythmic external input, and recurrent excitatory connections. So how can we ground these mechanisms in a conceptual framework to better understand their interactions? So the answer to that question is what we've been working on, and what we came up with is we can start by classifying them as resonant or synchronizing. So coming up on some of the experiments we ran, here we show the power spectral density of 6 simulations. The Nun experiment had no theta generating components with isolated pyramidal cells, no slow currents, and then direct unique Poisson input with no correlations. The other experiments activated one theta component at a time and then examined the power spectrum. The recurrent connections were activated in the plus RC, and then they produced a small bump in the 2 to 4 hertz range. Basket cells were activated in the plus BC experiment and then produced no spectral peaks. Routing input through the entorhinal cortex added correlations in the external input, and then shifted the power to low frequencies but didn't quite produce theta. Adding the OLM cells in the plus OLM experiment produced a robust theta peak, and then activating the slow currents in the pyramidal cells also produced a small but significant peak in the theta frequencies in the plus RES experiment. In figure B, relative theta calculations are shown. The plus RC and plus EC increased relative theta due to less specific increases in slow frequency power. So here we have a schematic of an experiment with the entorhinal cortex inactivated, and then input is directly arriving at the CA3 pyramidal cells. Both the OLM cells, basket cells, and the recurrent excitatory connections were active. In figure B, the power spectra of the 4 experiments are shown. The full experiment simulated both OLM cells and adaptation and pyramidal cells intact. The minus OLM was run with OLM cells inactivated. The minus SPYR had OLM cells intact, but adaptation and the H current removed from the pyramidal cells. And then finally, the minus both had both pyramidal cells and slow currents removed, as well as the OLM cells inactivated. The power spectra indicates that the theta activity persisted with at least one resonant mechanism intact, but then also interestingly showed that the pyramidal cells adaptation as a resonant component that may have interfered with the OLM pyramidal cell resonator. In figure C, we show the relative power in the theta band divided by the total power, and then normalized to the full, to the value of the full model run. Figure D shows the results of our procedures using constant current injections to cells to maintain a population average firing rate consistent with physiological data. which compensates for the variation in the firing rates resulting from inactivating the various network components from those experiments. Here we show that if you remove the resonant mechanism, theta actually goes up at low levels of acetylcholine. Here the recurrent connections and entorhinal cortex were inactivated, leaving the two synchronizing mechanisms in the model, the OLM cells and the BC cells. We tested three conditions. First were both the OLM and BC cells were active, and then with BC cells inactivated, and then finally with the OLM cells inactivated. Running the three conditions under three different cholinergic states revealed that different interaction modes between the two synchronizing mechanisms. In low levels of acetylcholine, they were equally effective at generating theta, and only one mechanism appeared necessary. With increasing cholinergic levels, BCs contributed to the contribution to the theta diminished, and then the high cholinergic states, they interfered with the OLM generating theta. And then finally, the OLM cells and the EC were inactivated, leaving the two synchronizing mechanisms in the model, the recurrent connections and BC cells. We tested three conditions again, first with both recurrent connections and BCs active, and then with BCs inactivated, and then finally with the recurrent connections inactivated. Again, running the three conditions under three different cholinergic states revealed a stable engagement of the BC cells in theta generation, while recurrent connections had a stronger engagement in lower cholinergic states. So again, I apologize for going from 0 to 60 on some of this content. I really wanted to get as much in here as I could. A lot of this is incredibly exciting to work with, and it really demonstrates the depth of what we do there at the lab. With that, I'd like to conclude. Also note for tools, techniques, and tutorials using the paper and throughout the presentation, check out scineuro.org. Again, if there was a lot of details we didn't have time to get over in 30 minutes. If you have any questions or comments, feel free to ask now. Or reach out to me via the email address shown on the screen. I can provide you with links to the slides and also the script for this talk if you want. Thank you all for your time.